or SOPs, are legally controlled documents used in regulated labs. They ensure that every person performs a task the exact same way every time. This reduces human error and ensures reproducibility. A key test trap is confusing SOPs with batch records. SOPs describe how to do a process, while batch records document what actually happened during production. Quality control, or QC, is responsible for testing products. Quality assurance, or QA, reviews the data and decides if the product can be released. A common trick question is switching these roles. Clinical trials follow a strict sequence. Preclinical testing uses cells and animals to assess safety. Phase 1 tests safety in healthy individuals. Phase 2 tests safety and dosage in patients. Phase 3 tests effectiveness in large populations. If problems occur at any stage, the process returns to research and development. That loopback is important. Environmental monitoring ensures sterile environments by measuring microbial contamination and airborne particles. This is critical in clean rooms used for drug production. Technical skills. Sterilization means destroying all microorganisms, including spores. Disinfection removes most but not all. This difference is heavily tested. Autoclaving is the most common sterilization method. It uses high pressure and heat, typically 121 degrees Celsius at about 15 psi. The pressure allows water to reach temperatures above boiling, which kills spores. 70% alcohol is more effective than 100% because water helps proteins denature. Pure alcohol evaporates too quickly and does not penetrate cells as effectively. PCR, or polymerase chain reaction, amplifies DNA exponentially. Each cycle doubles the DNA, meaning small errors or contamination also get amplified. That is why contamination in a negative control is a serious issue. Gel electrophoresis separates molecules by size. DNA is negatively charged, so it moves toward the positive electrode. Smaller fragments move faster through the gel matrix. Polyacrylamide gel electrophoresis, or PAGE, is used for smaller molecules like proteins because it has smaller pore sizes than agarose gels. ELISA works through antigen-antibody binding. It uses enzymes to produce a detectable signal, often a color change. A spectrophotometer measures absorbance of light. The blank removes background noise, so only the sample is measured. According to Beer's law, absorbance is directly proportional to concentration. Safety personal protective equipment includes gloves, goggles, lab coats, and sometimes respirators. The level of PPE depends on the biosafety level. Biohazard waste must be autoclaved or chemically disinfected before disposal. Sharps must go into special containers to prevent injury. Safety data sheets provide detailed information about chemical hazards, handling, storage, and emergency procedures. Always follow SDS instructions during spills. Biosafety levels range from BSL-1 to BSL-4. BSL-1 involves harmless organisms. BSL-2 includes moderate risk pathogens. BSL-3 involves serious airborne pathogens and requires inward airflow. BSL-4 involves highly dangerous pathogens and requires full-body suits in isolated facilities. A common test trap is airflow direction. In higher-level labs, air flows into the lab, not out, to contain pathogens. Biochemistry. Ionic bonds involve transfer of electrons, forming charged ions. Covalent bonds involve sharing electrons. Hydrogen bonds are weak interactions but are crucial in maintaining DNA structure. Enzymes are biological catalysts. They lower activation energy, meaning reactions occur faster without being consumed. Enzyme activity is affected by temperature, pH, and substrate concentration. pH is logarithmic. Each unit change represents a 10-fold change in hydrogen ion concentration. A drop from pH 7 to pH 4 means a 1,000 times increase in hydrogen ions. Buffers resist changes in pH. They are essential in biological systems to maintain stable conditions. Biological systems. The central dogma describes the flow of genetic information, DNA to RNA to protein. DNA replication occurs during the S phase of the cell cycle. Mitosis produces 2 identical cells. Meiosis produces 4 genetically different cells. Gram-positive bacteria have thick peptidoglycan walls and retain stain. Gram-negative bacteria have a thin wall and an outer membrane, which can make them more resistant to antibiotics. Understanding these differences is critical for lab identification and treatment strategies. Applied math molarity equals moles divided by liters. Always convert milliliters to liters before calculating. Dilution factor equals final volume divided by initial volume. A common mistake is reversing these values. Scientific notation simplifies very large or small numbers. Positive exponents indicate large numbers, negative exponents indicate small numbers. Significant figures reflect precision. Leading zeros do not count. Trailing zeros only count if a decimal is present. Beer's law states that absorbance equals a constant times concentration. If concentration doubles, absorbance doubles. Lab equipment. A centrifuge separates substances based on density. Heavier particles move outward faster under high speed. A volumetric flask is used for precise solution preparation because it has a fixed volume line. A pH meter measures acidity and must be calibrated with buffer solutions close to the expected pH. Glassware should never be dried with paper towels because fibers and contaminants can interfere with experiments. Scientific method. The independent variable is what the scientist changes. The dependent variable is what is measured. A hypothesis is a testable prediction. The null hypothesis predicts no relationship between variables. Experiments should include controls and be repeated multiple times to ensure reliability and reproducibility. Advanced concepts. A band in a negative control indicates contamination, not success. 70% alcohol is optimal for disinfection due to protein denaturation. Hydrogen bonds are the weakest but essential for DNA structure. During anaphase, sister chromatids separate and move to opposite poles. Qualitative data describes characteristics like color. Quantitative data involves numerical measurements. If absorbance is 0, concentration is extremely low or 0. SDS sheets provide critical safety information and must always be followed. Gram staining differentiates bacteria based on cell wall structure. Lab notebooks must include dates, procedures, data, and signatures to ensure proper documentation. End of deep study script. How to use this to hit 90%. Do this exactly. 1. Listen 2 to 3 times a day, morning plus night minimum. 2. Pause and repeat key parts out loud, especially math plus safety. 3.